Hi, my name is Madison Rogers, and I'm a graduate student in the Fantuza Lab at the University of Colorado Anschutz Medical Campus. And today I'll be describing my research briefly. In my lab, I study the very basics of craniofacial development or development of the face. We study receptors called PDGF receptors that when mutated result in cleft lip and palate. We're trying to understand how these receptors work in normal development so that we can better understand what goes wrong in craniofacial malformations like cleft lip and palate. So there are two receptors in the family that I study, an alpha receptor and a beta receptor. To track these receptors in the cells, I connected them to a green fluorescent protein as shown here. This system allows us to know where and when the receptors are inside the cell and when they're activated. Now in development, there are molecules floating around on the outside of cells that stimulate the cell and act as an initial signal that tells the cell what to do next. We call this molecule a ligand. The ligand on the outside of the cell causes two receptors at the interface of the cell to come together to form what we refer to as homodimers. Think homo is the same, dimer is two, so homodimer, two of the same. So here I'm showing two copies of the alpha receptor that have come together to form an alpha homodimer with that venous fluorescent protein, that green protein at the bottom, allowing us to track the alpha homodimers within the cells. I also generated cells that express two beta receptors together with that green protein in the beta homodimers. So once the receptors come together, they can perform a couple different activities. First, they can become internalized into the cell. So remember, they first are at the interface of the cell, then they can go inside the cell and move around to different compartments. I use the green protein as a tracking system as a way to determine differences between the movements of the alpha homodimers versus the beta homodimers within the cells. So here we have the green dots shown here are the alpha homodimers, and here are the beta homodimers. And I hope you can appreciate that the green pattern looks really different here from here. So this tells us that the alpha homodimers and the beta homodimers are moved around to different compartments of the cell from each other. So I looked at where the alpha homodimers and where the beta homodimers go within the cell by looking at a variety of different proteins within the cells to determine where these homodimers are located. Some of the things that I found are that the alpha homodimers shown on this top panel become internalized or go into the cell more quickly than the beta homodimers, which stay on the surface of the cell for a lot longer. So the second thing that the receptors can do within the cell is signal to other proteins. They can relay signals down a chain of multiple proteins, ultimately telling the cell to perform different activities. So in my studies, I found that the alpha homodimers and the beta homodimers relay those signals differently from each other, but that the beta homodimers are more dominant in the signals that they send off into the cell. And here, you can see this through the quantification of some of those signaling proteins. And we have the alpha homodimers in the green lines and the beta homodimers in the black. So you can see this more dominant signal from the beta homodimers. Now together, this movement of the receptors around the inside of the cell and their signaling relays to other molecules ultimately tells a cell what to do next, whether it should duplicate and form two cells and then more cells from there, or migrate to a new region. If we think about development of the human face in utero, there's a lot of growing and enhancing of certain features during development, hence the need for duplication and migration. So my research findings about the specific activities of these receptors using that green fluorescent protein allow us to provide valuable insight into how the human face forms during development. So I found in my studies that the beta homodimers, when they're activated, they lead to more duplication and more migration, probably leading to more dominant roles during development of the face. So this information can ultimately be used to better understand how mutations in the receptors can cause cleft lip and palate. Thank you for listening to my research today, and I hope you learned something.